mouse models, very important. There are several kinds of mouse models. The work that we do mostly is with patient-derived xenografts and with genetically engineered mouse models, and I'll tell you what these have really taught us. So patient-derived xenograft models is when either blood or tissue is collected from the patient and it's engrafted into a mouse. And when the tumor grows, it's really representative of the tumor that we see in patients. We can do drug studies in these models so that we really understand, because we can't take every drug or target into the clinic, right? We only have to take our best drug and best target into the clinic. So we can do these trials in the lab with these patient-derived xenograft models to understand toxicity, to understand the effectiveness of these drugs in these preclinical models. And genetically engineered mouse models are really, if you have a KRAS mutation, we try to mimic this mutation, like a KRAS mutation or a P53 mutation, into the mouse model so that we can understand the biology of this disease, and we can also understand what are the therapeutic vulnerabilities of that particular cancer type. So, I am also the director of the lung cancer PDX program at Mount Sinai. So we collect CTCs, biopsies, samples, and then we are engraft into these mouse models. And we can do different techniques. We can do histology, we can do whole exome sequencing, proteomics, RNA-seq, single cell sequencing, and drug response studies. And I know you're going like, whoa, what are these techniques? I'm going to talk about them in a little bit and what that has really taught us. But the point is, we can understand a lot about the disease from these mouse models. I'll start with the drug efficacy studies, and you will see a lot of these sort of dose response curves or tumor volume curves in this, any paper that you pick up, right? So this is with the PDX model, which was treated with two drugs, samatolisib and osimertinib, and we could understand that these drugs, the combination of osimertinib with samatolisib, if you look at the red line here and sort of the dark pink line here, if the curve is going down, which means the tumor volume is going down, which means that this drug is actually working in our mouse models. This is another example of that, where we worked with lurbinectedin, again with osimertinib, and we saw that if you see the red line here, that the combination is doing much better than this yellow line, which is osimertinib by itself. So really, mouse studies can really tell us that this drug is actually working with the other drug, either as a combination or even by itself without causing a lot of toxicity in the mouse. So that is the beauty of these mouse models. Again, this is another genetically engineered mouse model where we typically do immunotherapy studies, because these are grown in mouse that have intact immune system. So in this study, again, we found that anti-PD-L1, and this is a small cell lung cancer mouse model, where we found that, with ceralasertib, which is an ATR inhibitor, it works in combination with anti-PD-L1, and we are able to do a lot of downstream studies from these mouse models. And for immunotherapy studies, genetically engineered mouse models are really important. And again, we could compare what we found in the mouse with a clinical trials. So this is a proof of concept trial with berzosertib and with topotecan, and we were able to do RNA sequencing from these tissues. And the point is, what we found in the mouse was also confirmed in the patient samples. So this tells us that this drug is working in a similar fashion for the most parts. And this study, what that led to is to identify ATR inhibition or DNA damage inhibition as sort of an effective target. I'm not going to go into the detail of the biology, and I'm happy to answer those questions. But what that really, these studies are telling us is this is a drug that could actually work in this disease type, and it works by activating the immune microenvironment. And what that leads is to clinical trials, because that's our real goal, is can we take these drugs from the lab into the clinic? And what that study did is, support a lot of the clinical studies for this class of drugs, the DNA damage response inhibitors with immunotherapy for small cell lung cancer. Again, this is another example where we tried to mimic the maintenance setting with a drug called lurbinectedin, which is currently approved in the second line for small cell lung cancer. And again, this study told us that lurbinectedin could be an effective maintenance strategy for small cell lung cancer. And the beauty is, this is a trial, which is the IMforte trial, which is looking exactly at lurbinectin with atezolizumab in the maintenance setting. And our study is a preclinical validation that this combination could actually work in the clinic. And there was a press release which told us that this work could be, this combination could be effective for the patients with small cell lung cancer. We still have to see the data, but I think the point is that this is the pipeline that we want to follow, is take a drug from the preclinical setting to the clinical setting.